Welcome back to the second of three panels in the Genetic Age Symposium on Personalized Medicine. If the first panel was concerned mainly with trying to define the field and its implications and maybe make a brief forecast or so about the next 5 years, the second panel is concerned with painting a finer picture, a more detailed picture of the present and future environment for personalized medicine and genetic medicine. And by environment, I'm talking not just about the scientific environment about which we spoke some in the previous panel, but the economic and policy and social environment into which all of this wonderful new knowledge and eventually technology is going to disseminate. So here to help us think about this, we have a wonderful panel. On your left is Maureen Smith. Maureen is the clinical director of the New Gene project, to which Rex Chisholm referred here at Northwestern University. Sitting next to Maureen is Thane Kreiner. Thane is Senior Vice President in charge of Sales and Marketing at Affymetrix, one of our hosts today. Next to Thane is Kevin Schulman. Kevin is a professor at the Fuqua School of Business at Duke University and concerns himself a lot with health and health policy. Next to Kevin is Gerardo Jimenez Sanchez, who's the director of the National Institute of Genomic Medicine in Mexico. So as before, I will ask the panel to introduce themselves briefly, basically the same question, in order that the audience here and on the webcast can know where you stand. Tell us a little bit more about where you sit. Maureen, let me ask you to start off.
Well, thank you for the introduction, and let me just say that the first panel set the stage very well for me and made it very easy for me to tell you about what I do. As a clinical director for the New Gene project, this is a resource that we're developing here at Northwestern University in collaboration with Northwestern Memorial Hospital and our affiliates. And what we are attempting to do is set up a large cohort of participants who are patients in the Northwestern healthcare system and consent those individuals for genetic research. We obtain a sample from each of them and also obtain consent to allow us ongoing access to their electronic medical records so that we may associate their clinical information with their genetic information and do that in an ongoing manner. And so we hope that, and we have experience that this type of resource can provide researchers with the tools or some of the tools that they need in order to begin to study large populations. And while this is a hospital-based cohort, there's a lot that we're learning in doing this type of research. A couple of the things I wanted to also mention about providing or trying to do research on this scale is also the idea of using medical records as a way to develop a clinical phenotype and to try to understand clinically what's going on with a person's health care and with their health in general over time. And I think in this country it's imperative that we continue to look at medical records as a really important resource and a resource that needs to be more integrated throughout the healthcare system, throughout the physicians' offices that are out in the communities. And so this is, one way that we can begin to gather data and to look at that data and to use it for the kind of research that will make personalized medicine possible. And just one last thing I wanted to mention is the idea of recruiting research participants and how critical that whole endeavor is to this process. And until our country looks at the issue of passing legislation that makes it more possible for people to feel that they can participate in research, that their information is secure, that they won't be discriminated against, that the issues of their ongoing health care will not be affected by participation in research. And the idea of participating in research just for the idea of the future benefit of the whole, which would be a big cultural shift, I think, without getting results back or without getting results for a very long time. But that kind of cultural shift also needs to take place in this country in order for personalized medicine to take place.
How many people are represented in NewGene now, and how do you expect that to grow?
Approximately 6,000, and we hope to see that grow over time. It's, as I mentioned, because of the challenges that we're facing with just recruitment. I think if this non-discrimination bill passes next year, which I hope it will, that would make a big difference for us. So the kinds of changes, the climate in our country that will
actually, let me ask you. I just wanted the number to start with.
Okay.
We'll get into more conversation in just a bit.
Okay.
Thane?
Thank you, Robert. I'm from Affymetrix, and I come to this with a technology perspective. We make DNA chips. In fact, our CEO invented DNA chips in 1989 using technologies adapted from the semiconductor industry, which has enabled us to dramatically increase the amount of information that these chips can process. And so today, for example, we supply chips to academic medical research centers where they are genotyping using 500,000 SNPs, large populations that have been collected. The Framingham Heart Study is one set of phenotyped, clinically phenotyped, as Maureen was talking about, samples that is currently being genotyped using our SNP technology. The DNA chips are also very useful for looking at differences in genome function, kind of the weather map analogy that we heard in the last panel for what's going on with cancer. Is it going to metastasize? Where did it come from? What drugs is it likely to respond to? So the technologies, as I said, we provide to academic medical research centers, to pharmaceutical companies who use them for basic drug discovery as well as clinical trials. And we also provide the technology to diagnostic companies who are developing their own diagnostic products with our chips inside, kind of an Intel Inside model that we call Powered by Affymetrix. Robert introduced me as the head of marketing and sales, which is my current role, but I've been at Affymetrix for 13 years, and really the last 5 years I've been focusing on corporate affairs, which is the public policy and government relations perspective, and that's really one of my main interests in where I sit.
Thanks. Kevin?
Thanks. I'm a professor of medicine at the Medical School at Duke and professor of business at the Business School. And what I try and do is actually integrate those two different perspectives in the health policy realm, in the realm of cost effectiveness of new drugs and new technologies, and then also trying to balance that with the idea of incentives. One of the striking aspects of today's conversation already is a clear concern that people have about access to care. Access is directly related to the cost of care, and so if we can't reduce the cost of care, access is not going to get any better. On the flip side, we all want technology. The current standards in a lot of fields are that the technology doesn't serve all our needs or purposes. How do we create technology? How do we bring that to market? And then how do we do that in an affordable fashion? At the same time, giving people an incentive to do the research, to spend the money and see whether or not their ideas are going to play out. Related to genomics, we've been looking at a variety of different issues about personalized healthcare, the economics of personalized healthcare, and again, this relationship to incentives, and also how do we create a different environment that maybe could potentially reduce the cost of proving whether or not you're right. One of the things that we have to recognize is we talked a lot about associations already today. We've talked very little about validation and even less about prospectively determining the results of any of our hypotheses. And for $1 billion today, Pfizer got a wake-up call that, in fact, is critically important that we validate our hypotheses in actual clinical trials. And so, unfortunately, one of my roles is to constantly remind people that are excited about technology of that little last milestone.
Thanks. Gerardo?
Thank you. I am a pediatrician. I have PhD in human genetics and molecular biology and training in business administration. What I do right now is I run the National Institute of Genomic Medicine, which is one of the 12 NIHs of Mexico. It's an NIH devoted to using the knowledge from the human genome for the benefit of healthcare of the Mexicans. And so we are preparing a strong platform for what we consider a mid and long-term development for genomic medicine, which is mostly devoted to prevention and in part for treatment. We foresee that treatment, as we just discussed in the previous panel, will be probably the first steps towards a more personalized medicine, but really we're, in our mind, the major change in paradigm is with personalized medicine is prevention. So in taking some of the comments that we heard from the previous panel, we heard that the haplotype map is already available and that we can identify the major sources of human genome variation. But as we mentioned, there were four populations that were genotyped for the haplotype map, but none of them were from the Latin American population, which is a huge part of the population in the world, in Latin America and certainly in the US. So what we're doing, one of the projects that we are pursuing is to produce a haplotype map of the Mexican population as a proxy for the Latin American population. And this has, in our mind, this is a key element to prepare a solid infrastructure for genomic personalized medicine, because when time comes for clinical trials, stratify population, identify tag SNPs, and move on into more of the clinical arena, knowing the genetic background of this population will be important. And then we see, we begin to see that there is not only epidemiological but also economical interests in knowing the Latin American population. We very recently, actually a couple of years ago, signed a collaborative agreement with Governor Napolitano from Arizona. She's very concerned about the large Mexican population she has. And 3 weeks ago, Governor Schwarzenegger was in Mexico visiting. And one of the points that they were, the border state governors, interested on is in partnershiping in order to use this information for healthcare.
Good, thank you. The last panel kind of ended on a note that was talking a little bit about business models, and specifically mainly focused on the business models for the pharmaceutical industry. And if I can characterize it, the thought is that we move from an era of blockbuster drugs that are targeted to tens or hundreds of millions to narrower and narrower genetically segmented groups, possibly along the way, quote, rescuing some drugs that wouldn't have had sufficient response rate to merit being brought to market. I'd like to hear your perspectives on the impacts of this shift to personalized medicine framework, a personalized medicine set of technologies on other aspects of health and healthcare. And Gerardo, let me start with you to talk about prevention. This may be a little too simplistic, but as I understand, many of the studies of the cost effectiveness of prevention is that it's very hard actually to demonstrate its cost effectiveness. It's very hard to identify specific preventive interventions, maybe that's an oxymoron, where across the wide population the benefits outweigh the costs. Of course, where it has been demonstrated, there are tremendously effective preventive measures. Could you talk a little bit about the economics of prevention and how you anticipate genomics and personalized medicine will affect that?
So the first point that comes to my mind is that we have to realize that the way to individualize genomic medicine is really a long way. And we're talking about complex multifactorial diseases, and we're talking about several genes. We're talking about the SNPs in different regions of the human genome joining to confer some risk to high increased or decreased risk to certain common disease. And then a few minutes ago, Jim Caput from the audience was put in the context the environmental factor, which is really as important as the genomic factor. So this is really a long way down to identifying which are those SNPs and what are the environmental factors that confer risk to certain disease. When we're talking in the way about pharmacogenomics, that is to say, one SNP or two in one gene related to the response to a drug. So this is really very early stages where we were just discussing about. The goal would be to be able to say, well, these 12 SNPs will confer risk to this individual to diabetes or hypertension in the context of these environmental factors. So we have produced some economical studies, initial studies, in where we ask how much does the Mexican government invest or pay for complications of diabetes and hypertension only. And then if we were to analyze a set of SNPs and environmental factors, and we're able to push the appearance, the initial appearance of diabetes, say, 5 years, how much will this decrease? Then if we push it 7 years, 9 years, 12 years, what will be the impact in terms of medications for these individuals, in terms of the cost of complications of these individuals, talking about kidney transplantation, talking about losing a limb, talking about blindness, and so on. So how much will it cost? And we did a very careful analysis joined by McKinsey International, who helped us find out if developing genomic medicine for a middle-income country like Mexico was worth it or not. And in only analyzing hypertension and diabetes, it clearly shown that developing genomic medicine within the first 10 years, the amount of money that was required was measured in the context of millions of dollars. However, if we were to push the appearance or showing of diabetes, only diabetes, 5 years, that will be measured in thousands of millions of dollars. So it was a completely different scale. So it's certainly a wise investment just with these two common diseases.
That's I think, taking a laudable social point of view, saying that in the aggregate across a society, across an economy, in these two conditions, those cost benefits are positive. Kevin, let me ask you to bring that home to the setting in the United States, where over the span of 10 years that Gerardo mentioned, you might expect a typical employed person to change insurance coverage, 3 times, maybe 4 times. People that are uninsured may move in and out of Medicaid. A certain number of people will age into Medicare. Can you talk about how that plays out in our setting?
Yeah, I think obviously the great thing about the United States is we have a healthcare system no one else in the world understands or would want to adopt. I think if we start with the idea of prevention, I think we've tremendously underinvested in prevention, and I don't think even with this audience, we don't need genomics to do prevention better. We don't do well with treating hypertension. We actually don't even know what diet to recommend to the average American as we have this obesity epidemic. So there's some basic research questions that have nothing to do with genomics. Genomics might be nice interplays with them, but we can get the same benefit from preventing diabetes if we just got people to exercise a little bit more, and if we could figure out what we could recommend for a diet, if it's calorie or if it's amount of fat in the diet, we get the same financial returns. I think in terms of the structure for prevention, some of the issues that we're facing are the fact that, in fact, There are very little, if no incentives in the system. There's very little incentive on the individual. Generally, the individuals have no tools with which to deal with the system. You go to the CVS, you take your blood pressure, and you have no idea what to do with it. You have no idea if it's too high or too low. And so at the individual level, there's no teaching, there's no education, and then there's no financial incentive, although West Virginia is starting to experiment with this for compliance, get people who are diagnosed with hypertension to actually take their medicine. From the employer perspective, if you think about 10 years, very few employers today, large employers, may be in the marketplace in 10 years. The big three in Detroit are on the rocks. I was at something with MCI about 10 years ago talking about prevention. The people that were part of that executive team in that company, even though it's totally gone, what you have today is MCI is really WorldCom. And so from a financial perspective, if you're talking about 5 or 10 years to get a return from interventions in your workforce, there's very few firms that actually have a business plan that goes that long with the idea of the current workforce. And then finally, Medicare, we have this abrupt transition in the United States at age 65, or moving to age 67 and maybe 70s for the baby boomers, don't get too comfortable. But this abrupt transition, so when you do get expensive, if you don't die of sudden death, when you get your cancer or cardiovascular disease, you're going to be on the public's payroll rather than on the private first. And so there's a big incentive if your population is in the 60-year-old age bracket, instead of doing prevention, just cross your fingers and hope that they cross that threshold as quickly as possible. And so I think the way we've fragmented the system reduces the incentives for prevention at every single level, and I think that's a big problem to basic medicine, public health, and genomics across the board.
Now, Maureen, if I understand what Rardo and Kevin have just said, having a repository of genetic profiles linked to some characterization of phenotype through a medical record seems like a tremendously valuable asset to have in this market. Can you talk a little bit about the business model for a biobank? Now, I understand that you're academically linked, but there also, I know, are private sector biobanks that are out there. There are full-fledged national biobanks like the UK biobank that Rex referred to. Can you talk a little bit about that business and how you expect to see that evolve?
So I think the business of biobanking, one of the things that we need to see is a lot more collaboration among the investigators who are involved with biobanking, as well as the medical establishments who have access to patients. And whether that is through commercial enterprises or within, such as NewGene, within a hospital-based system itself, we need to focus on how we can really build these resources that have long-term results and that don't have short-term return. And I think those are some of the things that are very difficult in our healthcare system to bring about. And also, I think part of that collaboration involves incentivizing those who are directly involved with the medical records, who are inputting the information so that it's clear and accurate, and so that what we take out of it is represents a true phenotype of a participant. And so I think these kinds of activities and collaborations are really critical, and not ones that have routinely taken place within our medical establishment. Beyond that, I think that the other part of this, which I can't speak to in great depth, but which I think is important, is being able to provide those samples, those phenotypes, that information that's in a biobank to investigators in a way that they can access them reasonably. And so we can't make the costs of accessing samples so excessive that they can't use them. And so there needs to be mechanisms developed that can make these samples and these banks accessible to investigators across the country.
Good, thanks. Thane, one of your client sectors for Affymetrix is the diagnostics industry, an industry that I may be too pessimistic in saying that it's sort of been marginalized, at least in this country at times. It has not, for instance, enjoyed the same growth that the pharmaceutical industry has had. What's your sense of the impact of personalized medicine on that industry and its business model?
Well, I think there's the potential for a very significant impact, but it will require some changes in policy and regulatory frameworks. And let me give you just a couple of examples. The two areas where people usually talk about personalized medicine being applied first are cancer and pharmacogenetics. So in cancer, Brian gave us the example of Herceptin and HER2/neu. It's a single marker. The cost of the drug is very high, but whether or not the drug is going to work is determined by a very simple laboratory-developed test. How do we determine what the appropriate value is to ascribe to the drug versus the diagnostic? If you have a drug that's thousands or tens of thousands of dollars a month, how much is the diagnostic worth to tell you whether to use that or not? And I think that's one of the things that the diagnostic industry has been grappling with, struggling with sort of 14% operating margins versus 60 or 70% operating margins in the pharmaceutical industry. Another good example that was also brought up is pharmacogenetics, and there's already a pharmacogenetic test that's been cleared by the FDA. It's made by our partner Roche, the AmpliChip P450, and it looks at a number of alleles or genetic differences that are involved in metabolism of commonly used drugs, neuropsychiatric drugs, cardiovascular drugs. Now, this gets back to Kevin's remark about the time frame for analysis. Almost everybody is going to take some class of drugs that is metabolized by the P450 enzymes during their life. Would it make sense as a matter of national health care policy to take a genotype for everybody when they're born? And that's 4 million, 5 million live births in the United States per year. The cost of adverse drug events per year, there's a million to 2 million adverse drug events and 100,000 deaths, is certainly going to be much higher than understanding the drug metabolism differences. So I think there is a huge opportunity here, but it requires some policy changes. And I also want to echo what other people have said about being careful about genetic determinism. It is true that genes can be identified that are very much related to how you metabolize drugs, but even with something as simple as that, there are other factors. Did you drink grapefruit juice? Are you taking other drugs that are in your environment? So we have to be careful not to say we're going to have a genetic test that will tell us whether we're going to get cardiovascular disease or cancer or Alzheimer's disease and drug metabolism, and that's the end of the story. And I think that has to be worked out as well. And that's part of the deciphering that people have talked about with these large biobank projects.
Well, you raise, I think, a very important question, which has to do with the regulatory environment for all of this. And I'd like to spend a little time just hearing from the panel your take on the openness or not of the regulatory environment as it exists today, if there are particular regulatory changes that you think we're likely to see or are necessary. And Maureen, let me start with you because when you mentioned linking genetic information to an electronic medical record as a representation of a phenotype, all of my Hipaa bells started ringing like crazy. And if you could talk a little bit about Hipaa and how you navigate those waters, I'd appreciate it.
Well, so that's a very good question. Hipaa is a very complicated rule, and we do our best and work with our legal systems here to manage the Hipaa issues. I think one of the things that I did mention that might help is that when we provide information or DNA samples and clinical information to a third-party investigator, we have to provide that information in a de-identified manner so that that person doesn't have the set of identifiers linked back to them. And so the investigator does not have knowledge of who that person is. we have a number of agreements with the clinical entities that we work with. We are part of the university, and we work with clinical entities such as hospitals in order to collect samples and consent participants. And so we have to have agreements with those different clinical entities that allow us to conduct our research and to obtain the information back from their medical records and scrub it of identifiers and then be allowed to provide it back to investigators who wish to use that information, those samples. So it's a fairly complicated process of working through a number of issues to protect participants. And that's not to say that we don't want to protect participants. I think that is utmost and foremost in our mind is protecting their privacy. And so working with Hipaa and with our clinical entities and the various laws that also add on to regulations that we have to abide by in our own state, working through all of those issues are critical so that we are protecting people and that we are providing information in an appropriate manner.
Could you talk a little bit about how you balance out some of the conflicting priorities, and in particular, how inclusive the consent form is? If I understand how most consent forms that are approved by IRBs are, they're relatively narrow, that say that, well, this sample is consented for use in this research project and for that purpose. When I speak with patient advocates about biobanking, what they say is, well, wow, if we can contribute to the advance of knowledge in any way in our field, we'd be delighted to have more of a blanket consent for our sample. Not all of them are quite that open to it, but some of them want some economic return from research on their own sample. But generally, the sort of mood that I get from patients is, yeah, give me a blanket consent. I'll get everybody in my patient group to sign up for it. So how do you navigate? How open-ended is the consent form in NuGene?
So for NuGene, it is very open-ended in terms of participants consent to allow their information, their sample to be used for broad-based genetic research. And part of the reason I believe that they consent to that is because they're not identified during the course of research. And however, it's interesting to hear your comments because there is a fairly large debate across the country and around the world about consenting for each use of the samples. And in many cultures and parts of this country, they believe that it's very important that for each use of those samples that patients should be given the opportunity to consent. And so it is, we have chosen a broad-based consent because our samples are provided in a de-identified manner. But I think that there's some controversy around that issue.
Gerardo, how is that handled in the Institute for Genomic Medicine and the research that goes on there?
So we have a biobank as well that includes so far about 2,000 Mexicans from 7 different regions of Mexico. These are anonymous samples, and therefore we have a wide consent from our IRB. And when we have to keep in context that this is a country, although it is our next door neighbor from this country, is a country with a different educational level average. So we have to be very clear on how we explain what genomic medicine is, what the impact of this is. And the reason I'm saying this is because in a few years' time, when we talk about global markets, we really need to prepare our market if we want them to understand what we're talking about when we say we need to determine some SNPs and link this into a risk and give you a more personalized medicine. If you go down South America and they've never heard what a human genome is, but they've heard about cloning and embryos and this and that in wide Catholic part of the continent, this will be just chaos. So we're really working very hard in preparing what will be our major market. And so what we do is that we have sampled so far 7 states in Mexico. We just chose north, south, east, west, 2 in the center, and 2 in the U.S. border. And what we do is that we ask and have, we are very active into community engagement. So when we do the sampling, we engage from the governor of the state, the secretary of health of the state, the president of state university, and with the institute, we all together go to the community through the media, through different open discussion sessions, at least 5 per state before we do the sample collection. And one thing, the point I want to make is that the consent form, we prepare it in the form of a poster, and we post it through the university and through downtown and different parts of the capital city, so 15 days before we collect samples, so that people can read freely on what this is about. And our consent form is in a format of 12 easy questions dealing with IP, dealing with this risk of discrimination, dealing with anonymity, and so on. So that is one part of it. The other one is the open discussion session, so that they read, they can come and ask, and then the media is the third component. So we really are very open into informing the population what this is about. And usually when we collect the samples, which is only once, we invite people for the HAP map, and we usually collect about 200 individuals, that is about 1,600 vacutainers in about 3 hours in one state. So really people are very active and willing to join in. But the point is that we made a wide consent form, and we asked for permission to use this for different studies.
Thane, our regulatory mechanism for approving medical technologies seems to be pretty well adapted when the medical technologies remain pretty stable. You find a chemical entity, you demonstrate that chemical entity has a biological effect, it's approved for that indication, and then docs prescribe it for whatever they want to. That's another story. But if I understand some of what you're saying about genomic technologies, those seem to be evolving pretty rapidly, that we're finding different constellations of genes that have a given effect. We've got a lot of hypotheses. How is that regulatory mechanism, how well is that regulatory mechanism adapted to genomics, and what sorts of regulatory changes, if any, do you think would assist in the dissemination of genomics?
Mm-hmm. Well, I think one of the big differences with genetics and genomics that we've been talking about today is the concept that you're looking at many things at once versus a single piece of information. And moving to what FDA is calling multi-analyte test is one of the regulatory paradigms that's under consideration. However, I think there's some nomenclature issues here, and it comes back to the weather map metaphor that was used in the last panel. If you're looking at a constellation, as Robert said, of genes that are expressed differently in tumors that metastasize versus tumors that don't, are you really looking at 1,000 different genes, or are you looking at, does the tumor metastasize or does the tumor not? And is that one analyte or is that 1,000 analytes? And the hurdle to look at 1,000 different pieces of information, even though taking a temperature in Saskatchewan may not be that important if you have the temperature in 4 other points to know it's going to be cold in Chicago. So I think there's a lot more effort that needs to be put into evolving the regulatory paradigms. My sense is that in the U.S. at least, the FDA is very open to that, has really engaged with industry and with academics to understand the changes that are being brought about by the ability to look at large amounts of genetic information. I also think that the kinds of legislation that Dr. Hughes talked about earlier this morning are necessary, and we in fact endorse the Obama legislation and are very hopeful that will move forward. I wanted to make a comment, if I may, on the GINA Act too, which Dora also mentioned. We have been supporters of that for about 5 years, and I think one of the core issues in understanding the Genetic Information Non-Discrimination Act for us is that privacy, that the cost of absolute privacy, and there was an article in The Economist just last week for other kinds of information on the internet, is extremely high. And guaranteeing absolute privacy is probably not something that is economically feasible for medical information or financial information or any other information that you really care about. So what we really want to do, in my view, is protect against the misuse of information. And that's what GINA does. And we don't want people to be discriminated against based on their genetic information, their predisposition to diseases, or how they're going to metabolize drugs, whether that's in their health care or in their employment.
Kevin, anything to add in the regulatory frame? Or I might also ask you to comment on the reimbursement frame as well.
I think on the regulatory, you're seeing a couple of different things. So on the FDA, there's this guidance about pharmacogenomics and submission, where for a couple of years, the FDA has asked manufacturers to submit data on the genetic experiments they run, so FDA can see what the data look like and begin to understand how those data might be useful in improving a drug or regulating a drug. Obviously, things like SNP testing, P450 testing, have been a part of the label for some time now. On the device side, we have an interesting paradigm with the draft guidance that FDA is putting forward, both on testing itself, the difference between evolving a specific set of regulatory parameters for some of the gene expression testing, which basically is how do you regulate mathematical algorithms. So that's a real question. And then out the relationship between that and CLIA, the framework by which most of these things have come to the market. And so, to be frank, there's no issue on the research side. This is much more of an issue if we think these things are ready for commercial application. There is a debate about the appropriate regulatory framework in that space. Once you get to reimbursement, I was part of a conference a couple weeks ago that was a pretty stark conference in terms of the ability to actually get insurers and Medicare to pay for these things. If you think about the excitement about this technology, there's actually a whopping 1 FTE person, full-time person in Medicare that deals with diagnostics, and then they said the entire diagnostics effort in Medicare is 3 FTE. And so, that's not going to be an environment that's very responsive to exciting new changes in technology. On the private payer side, I think you're going to see a very low bar to adopting things that may not be scientifically rigorous, but hold the promise of reducing costs. And you'll see a very high bar to adoption of things that are going to be more expensive. And so out of Duke this summer, we had a very exciting paper about prognosis for patients with early stage lung cancer. We don't know what to do with people who are high risk for early stage lung cancer. There's no trials of drugs that whether or not these people, whatever makes them high risk for early stage lung cancer, make them entirely refractory to whatever chemotherapy regimen we apply to them. So it's not really clear what the economic implications of something like that might be. And a risk of the economic implications of figuring out non-responders were much more straightforward.
Your comment, I think, leads to a pretty important issue here. If personalized medicine, if genetic medicine follows the trajectory that almost every other medical technology has followed, at least in the United States, it will be applied to the elite first, to those who are most able to afford it, and then trickle down, to use a phrase from an earlier era, it'll disseminate more widely into the rest of the population as either its cost effectiveness is demonstrated or the social pressure for equity is exerted. Is this move towards personalized medicine likely to follow that same trajectory, and is there the risk then of augmenting health disparities because of it, or can you imagine a model in which it disseminates in a way that doesn't augment disparities?
Is that...
That's for anybody who wants to jump in on it, and I'll ask everybody to comment on it.
Yeah, I think there's no question that the first market of this might be a cash market, that people who want this information are going to go buy it. To some extent, that's not necessarily a bad thing. It depends on whether you think the technology is ready for prime time. And if there's a lot of uncertainty, the people paying the most to get this information could be really interesting guinea pigs, which wouldn't be a bad thing compared to most of the research that we do, which is the other way around. But I think there are a couple of things that are going to exacerbate disparities related to the technology. And the first, so we talked about the technology and accessing the technology, but the technology is only useful if you have access to some type of electronic medical record that's portable. And in fact, if you think about who's going to have those first, it's the same population. So I think there are a couple of things that are going to be barriers that are going to exacerbate disparities out of this. And I think that's going to be the role of the public programs to figure out solutions to that. Medicaid's not a very stable platform to offer people prevention or even information technology, because people moving in and out of that population, obviously the working poor who are increasingly uninsured in the country aren't going to have access to either the system or the infrastructure that will manage their records or their information. So I think portability is key if we're going to try and work on at least the information accessibility component of the disparities.
Gerardo?
So, once again, this was a major concern when the genomic era came up for Mexico. So whether we develop, question was, should we develop genomic medicine for the Mexicans, or should we wait until the U.S. develops it and we just import it? So that was a strategic question, because developing genomic medicine for the Mexicans, really what matters in terms of not only of an economical impact, but also on the social impact. What matters when new medical technology arises is the timing. So timing is key. When a country develops or applies emerging medical technology to its population, the World Bank has shown with very clear examples such as vaccinations in the 70s and 80s, that when a country develops or applies emerging medical technology on time, it helps reduce the gap between the rich and the poor, and I'm talking about richer countries, poorest countries. Now, it also has an impact within the country, which will be much more dramatic than the one that could be in this country, because the poorest are poorer in Mexico and the richest are richer in Mexico. So, and this is just once again an example of what a pretty good proxy for many countries in Latin America. So, so one of the strategies that the Mexican government did in order to at least buffer as much as possible these risk of differences between the rich and the poor was actually to create a National Institute of Genomic Medicine, that is the federal government regulating a national platform for genomic medicine. So that was the very first bill that was passed through the Mexican Congress, creating this NIH. The second bill related, and I just wanted to touch on this, the second bill regarding genomic medicine in Mexico is actually already passed the representatives, and it's already at the Senate, which is non-discrimination. And there is a third one coming up already in the lower chamber, which is a genomic sovereignty. That is, who has the rights in IP towards the Mexican genomic biodiversity, which is really, in terms of economy towards the future, it's a key issue to decide whether it's worth for the Mexicans to invest in genomic medicine or not. So I just wanted to conclude saying that the Mexican Congress has several bills regarding genomic medicine in the works, 2 of them already out, some of them in the works, that will help create this environment. And just as a final comment, to really make the point that education at different levels, not only in Mexico, here, everywhere, in terms of genomic medicine, we really need to get docs to know what this means, what to do with the P450 panel shows this way or this other way, how to prescribe this or that. And not only medical doctors, but society at large need to be educated formally along the lines of genomic medicine and its impacts.
I anticipate that in our next panel we'll dive into that topic in some depth. Thane, anything to add?
A couple things. again, we're a technology provider, so we have sort of unique and central position, you might say. And one of the things that we've thought about a lot and that concerns us is that it's not just the ultimate diagnostic tools or personalized medicine tools that are developed using our technology or any other technology and how they might be applied in ways that heighten differences in access to medicine, but it's actually what kinds of information you look at the beginning. And if you just look at certain populations, as was brought out a couple times today with the HapMap, instead of looking across all the genome and across all the populations, you might not find all the variations that are important in different human conditions and traits. So the studies that are done actually in the conditions and diseases that affect different populations tend to vary by income, as most people know. And so if you think about infectious disease work and susceptibility to very common diseases in Africa, more of that should probably be done from our perspective than is being done, whereas there are a lot of studies, rightfully so, I think, given the high cost on more developed economies on obesity and cardiovascular disease. So I think this idea of access actually runs the whole spectrum through from the research and the deciphering of how genetics relates to the human condition to the ultimate application in the clinic.
Yeah, if you applied it according to the global burden of disease, it would have a different profile than we have now, which maybe more resembles the global ability to pay. Maureen, anything to add?
Just a few comments. I have practiced as a genetic counselor for almost 20 years, and I think one of the things that's most striking to me is that there's always been a huge health disparity, or a huge disparity of who gets access to genetic tests. And one model I would hope that possibly personalized medicine may follow is that of something along the line of newborn screening, which is available to every baby that's born in the United States. And I think if we're able to integrate the advances in personalized medicine with the public health system, then we have opportunities to offer testing and provide the education to a wider range of the population. Good,
thank you. Audience, let me invite your comments and questions. And I guess we've got a mic runner. You've got a mic there. I'll hand this mic off to somebody. Going to run one on this side? Yeah. Right here in the middle. I'm sorry. Right here.
First of all, I have to thank the panel. This is a remarkably interesting panel. I'd like to direct a question to Ms. Smith.
Could you keep the mic pointed right towards your face?
Okay.
Thank you.
I wear a couple of hats. I'm a lawyer, retired. I'm a senior, and I'm a patient at the hospital in one of your departments. So I'm a prime person to be in this audience. And also I'm a patient in the Cardiovascular Institute. This is the first I've ever heard of the New Gene Project. And I've been a patient for 4 years. Number next, I'm troubled by the guarantee or the pseudo-guarantee of the de-identified genetic coding, our genetic information. Because when one goes into this hospital, Northwestern, which is marvelous, or any other hospital, you're asked, the first thing you're asked is your social security number, and then you are asked to sign, of course, a privacy form, but you know that that information is going to get out because there are non-professionals that are handling your information. So I wish you would say something about that.
Actually, if I may, let me ask her to comment on the first set of questions.
So all I can say about you not hearing about the New Gene Project is it's unfortunate. We have worked to integrate into a number of different departments and throughout different areas of the hospital. But that's a process that is continuing and is not smooth because our framework for doing large-scale research has not been done before. And so we're setting up new systems and learning our way. And I hope that in the near future you will have access to deciding whether or not this is something you would be interested yourself in participating. So I appreciate your comment about that. And secondly, regarding the privacy issue, the way that NewGene operates, and I understand your concerns, and I hear them, we hear them all the time. And that's one of the reasons why that is an issue that I have to go to whenever I speak about this project, is patients' concerns about privacy. In our system, we have a dual database system where the information that is identifiable about a participant, and we maintain a minimum amount of identifiable information on a participant in a database so that we can recontact those who have agreed to recontact for a variety of reasons. And that's a database that is separate from our actual database where we would hold any clinical or any genetic information. And so they're maintained separately and linked by a code. And so in a sense, if someone were to break into one or the other, they would not be able to link the two and to link up any data from one to the other. And that's how we operate in terms of trying to provide some protections to our participants. Again, as I said, we do scrub the clinical information that we get from the medical records. It's scrubbed of identifiers and not maintained with any of the genetic information about a participant.
I should point out that there are two social goods kind of fundamentally in tension here. On the one hand, the social good of maintaining the privacy and confidentiality of an individual's information, which if you take it to an extreme, says, well, let's really keep that information isolated from all other information. Maybe like in a paper chart, the way we do it in most of medicine today. On the other hand, there's the social good of research, which fundamentally is a collective enterprise where sharing and mixing of the information is necessary. And so the question is, how do we balance those two social goods, or how do we balance the social ill of the risk of releasing some of your personal information versus the social ill of research not being conducted? Let me open it to another question. There was a hand. Yes, right there in the middle.
Yes, I wanted to ask Maureen to comment a little bit more about the lessons learned in newborn screening paradigms. These strike me as a very useful model for thinking about broader applications of genetics for the population, both in terms of which disease entities one would consider, the ability to use that information to modify the course of a disease, the economic impact, and then how you actually begin to apply this at a practical level.
So in newborn screening, the kinds of conditions that have traditionally been screened for are those in which there's some kind of treatment available. And I think that's a good model to think about with personalized medicine is, are we looking at conditions, common, complex conditions that are actually where we can provide some kind of prevention or treatment? And the way that newborn screening is done is there is a test that's run on a newborn with a heel stick in the first few days of life, and results come back fairly quickly, and it can be confirmed within a few weeks of confirming whether a child actually has a genetic condition or not. And so the treatment can be instituted very, very quickly. And so the beauty of that is the response system, I think, and the diagnosis, which can take place so quickly. And I think this is a perfect kind of thing to think about. I'm not sure that the newborn period is the perfect period when we think about adult onset diseases, but for some, maybe that's a model that would work in terms of thinking about lifestyle changes and modifying those kind of factors. So, I think that it is a possible model that is interesting. The economics of it is that the costs are spread out amongst everybody having a child or throughout the state. And depending on, they're basically run on state-by-state level. And how costs are incurred, it's generally a minimal cost that is tagged along with the birth costs. And so they're fairly small and are equalized across everyone having a baby.
Any other panelists care to comment on the newborn screening question? Any further questions in the audience? Yes, right there.
As you indicated, it's determined on a state-by-state basis, and the number of tests that will be run in the newborn screening panel. And my question goes to how big a car do we want to design? I was recently at Harvard for a 2-day conference on personalized medicine, and the question was made that sometimes you don't need the most fancy Bentley. Sometimes the Toyota is what is most effective because it does the best for the best number. So in that line, the issue becomes, in terms of return on investment, what is the evidence in terms of validating studies either for a diagnostic or for a therapeutic, given that our clinical trial system is actually quite faulty, and there have been many problems with fast tracks, and certainly as the Dr. Schulman mentioned, and we had a curbside earlier with the Pfizer data, and we can name several other blunders that have occurred in the last 5 years. So if we're going to get this right, hit the medium, do the best for the most common good, what needs to be done in terms of our infrastructure to change our clinical design structure and put excellence back in, and excellence at a quality level that we can all afford?
Thane?
Can I comment? Yes, I think it's a great question, and I think there are a number of different ways one could start addressing that, but a lot of it comes back to me to the intended use. And so some of the patterns are going to be very valuable, and we want to fast track them because there's no other way to get an answer about whether a breast tumor is going to metastasize or not. And that's very useful information. Even if the test isn't 100% accurate, it's better to know with 90% confidence than not to know at all. On the other hand, and this was brought up earlier, there are some common disorders, obesity, cardiovascular disease, where what we know today is that a lot of genes seem to be involved, and the relative contributions of each versus the environmental contributions are really not well understood. So maybe the most effective public health measure there is to do what we've been doing, and the question is whether it works or not. Don't smoke, don't eat or drink too much, and exercise regularly. And that may be the best public health solution. So I think it's not going to be one size fits all, just as we said about drugs, for the regulatory pathways or the clinical trials. I think we really have to look carefully at how the information is intended to be used to improve the patient's health.
Kevin?
Yeah, I would say we're at an early stage of really thinking through this problem. I mean, if we decide that this technology is really one that has an opportunity to change the trajectory of the path of medicine, then we need to build an infrastructure to test that hypothesis. And we need to test multiple hypotheses on big populations of patients. We talked about 2 million people in a longitudinal epidemiologic study, but our calculations just for cardiovascular disease are probably you need about 500,000 people at least with diseases to look at low frequency interactions. So I mean orders of magnitude greater than what we're doing right now as a research infrastructure so that we can get the answer, get it expeditiously and bring it out. And I guess I'll take a point with this idea with breast cancer. It's not anything against any specific test. I was having an argument with my sister's actually training to be a dietitian. And I from putting on my ClinEpi hat, I explained to her that actually most of these very small studies that she's researching and looking at basically are probably wrong. If you see a study in the literature and there's 10 or 15 people in there, That means that they had a positive p-value. There was another equally likely study that never got published that was negative. And in fact, the issue we're facing right now is we have a lot of small studies. We see the ones with positive p-values. We never see the ones with negative p-values. We don't have a chance to confirm them. And so this idea of a rush to market because I have something with a positive p-value is really problematic. I'd rather have a rush to confirmation, because if I can cure breast cancer, if I could change the prognosis, I want to do that. I don't want to wait 7 years to figure out whether we're right or wrong. On the flip side, I don't want to repeat what happened with estrogen, which is it took women 25 years to find out that, in fact, the observational studies we couldn't validate them, at least in the clinical trial setting. So if that's where we're at, then we need to get serious about it and have an infrastructure to test it. On the flip side, that might make the technology development piece cheaper. I wouldn't say it's going to reduce the price to consumers, but I think what it would make is it easier for firms to get into this business and have an opportunity for having more tests and more competition, which might change the trajectory of this market over time.
Panel, let me ask you the same closing question that I asked the first panel, which is to ask you to put on your forecaster hats briefly and think out 5 years to the closing month of 2011, and particularly in the domains that we've been talking about here in terms of regulation and access that people have to this, and also particularly maybe this last question that was asked about research methodology and research infrastructure. Where do you think we'll be? Maureen?
I was afraid you were going to ask this question.
So you had plenty of time to prepare.
Yeah. I guess that I am looking forward to some legislation that makes some of the research possible, passing in the next 5 years, and maybe providing the groundwork for a national biobank in this country. And I hope that the regulatory framework for allowing that to occur, or allowing multiple biobanks to work together in this country, will be at a stage where we actually can collect significant data. And so I think that's probably as much as I'm going to forecast at this point.
Yeah, let me just point to a particular couple of words you said there, which is multiple biobanks working together, and I think that's a key, isn't it? Mm-hmm. Thane?
Well, I think there's no question that there's going to be a huge amount of genetic information. People are generating billions of genotypes every week now using the technology, and it's becoming more and more affordable to do very large studies with well-phenotyped cohorts, such as the one that Maureen talked about in the Framingham study that I mentioned earlier. I think we will begin to see some of the informatics deciphered out of those studies within 5 years, and some of those findings will be applied to development programs for new therapeutics, and there probably will be some cases where the combination of genes affecting common diseases is sufficient to warrant some kind of intervention. So on the genotyping side, I think that we'll be in the thick of it, but not all the way to the point where we can have everybody have their own gene chip and share that information with the doctor, and the doctor know exactly what to do. That's probably 20 years away. I want to come back to Kevin's comment, though, about cancer profiles as another prediction, because I think it's a very important one, and I agree completely with Kevin. And in fact, there are about 18 trials going on right now with different cancer signatures, so I expect a number of those will be available within the 5-year horizon for people who have cancer, and they will be validation studies that confirm the use of that pattern to differentiate who's going to respond, or how the cancer is going to progress, or what kind of cancer it is. So I think that is moving forward, and I think we'll begin to see the benefits of that within 5 years.
Thanks. Kevin?
I think gene expression is a really interesting idea. It's totally agnostic, right? You don't have to know anything about genetics. You just have, green or yellow or green or red dots on your chip, and you add it up. and you come up with a prediction score. And so it's interesting as we take this technology from those who are really detailed scientists to the more clinical applications, you know, how you're going to put that together is not how we would have predicted a couple of years ago. And so one of the things we have to do is make the technology accessible to a lot more people to think about how we could use this and use this creatively to get answers for patients, which might not be the most elegant biological solutions. The systems biology piece is going to take us decades to answer, but I think there are uses and useful pieces of data today. I think the pharmacogenomics, I'm a little cautious because I first did a P450 slide, I think on the first maybe when Affymetrix first started, and we still don't have P450 accessible for everybody writing a script. But I think there are hints, both in some of the drug metabolism stuff and especially in oncology, that we're going to make some fairly significant progress over the next couple of years.
Gerardo?
Well, I agree that in the next 5 years we'll see a dramatic increase of genomic data coming up, which is really being produced all over the world day after day. Again, I see that bioinformatics will let us identify some risk-related genes. But one of the things that I hope to see is that we realize that in the same way that knowledge-based economy is growing by sharing data, by doing synergies and creating actually the data mining. Same thing we will do in the research arena, which is realize that we have to join forces, namely in biobanks, bioinformatics, databases, and those things, with all the ethical and legal issues taken care of. But the fact that we really need to join forces so that we can make a real impact. I don't foresee that in 5 years we will see a major revolution as to how we conduct regular healthcare. However, I share the vision that pharmacogenomics will be probably the first part of the genomic medicine to be on board, and I agree that cancer is the area where we most likely will see progress in genomic individualized medicine.
Good, thank you. Before we thank the panel, I'd like to say what's about to happen next. We're going to move right into the third panel. So that means third panel panelists, if you don't mind coming right down, and sound folks, if you've got people ready to make the mic changes so we can make this happen quickly. With that, I'd like to ask all of you to join me in thanking this panel for a wonderful job. Thank you so much.